In this video tutorial, I am going to discuss about gluconeogenesis. Let's first see what is gluconeogenesis. So, if we dissect this word, we get gluco, that means glucose, neo means new, and genesis means synthesis. So, the literal meaning of this word is new glucose synthesis. So, of course, in this pathway, glucose is synthesized, but it is synthesized from the where? It is synthesized from the non-carbohydrate precursors. These various non-carbohydrate precursors, they undergo sequential enzyme catalyzed reaction to give rise to glucose. What are these non-carbohydrate precursors? In our body or in human, there are total 4 different types of non-carbohydrate precursors. The first precursors are the glycolytic end product. These are pyruvate and lactate. Second is the glucogenic amino acids. Glucogenic amino acids. Remember, in our body there are total 20 different standard amino acids, and out of these 20 different standard amino acids, 18 amino acids are glucogenic amino acid, whereas remaining 2 amino acids, these are leucine and lysine. These 2 amino acids, they are non-glucogenic amino acid. So, we can say that all standard amino acids are glucogenic amino acid except for leucine and lysine. The third important precursor is the propionyl coenzyme A. Propionyl coenzyme A. And the last one is the glycerol. So, these are the precursors which can give rise to synthesis of glucose. If we talk about the location of gluconeogenesis, the most of the steps of gluconeogenesis, they occur in the cytosol, whereas initial few reactions sometime occurs in the mitochondria. And if we talk about the tissue level, 90% of the gluconeogenesis, it occurs in the liver, and remaining 10% gluconeogenesis, it occurs in the kidney. And in the kidney, it specifically occurs in the renal cortex. And there is one more tissue in which gluconeogenesis occurs, that is epithelial cells of small intestine. Now, in these epithelial cells of small intestine, very negligible amount of gluconeogenesis occurs. Now, this 90% and 10% division of gluconeogenesis, this occur during a routine fasting condition or short-term fasting condition. When the fasting is long-term, at that time kidney increases its capacity of the gluconeogenesis. So, in the long term, it increases from the 10% to the 40%. So, accordingly, percent-wise, liver's contribution decreases from the 90% to the 60%. So, remember these numbers of the gluconeogenesis, these are for prolonged fasting condition, whereas these numbers are for the short-term fasting condition. This we have to keep in mind. Now, let us discuss about the significance of gluconeogenesis. Significance. The first significance is that as we know that in gluconeogenesis, glucose is getting synthesized. So, this pathway, it can be considered as an important source of glucose. So, it can act as a source of glucose. See, during different times, different substances can act as a source of glucose. When we had just taken food, at that time that food will act as a source of glucose for us. Once you are not taking food and 6 to 8 hours of the fasting is there, then at that time glycogen will act as a source of glucose. Now, once glycogen gets depleted and our fasting becomes little bit longer, we don't take any food, then at that time only gluconeogenesis will be acting as a source of glucose. The second important significance is that in our body there are certain tissue which fully or exclusively dependent on glucose for the energy purpose. What are these tissue? Our brain, retina, red blood cells, renal medulla, cornea, testes. These are some of the organs or tissues which are fully dependent on glucose for the energy production. And if your glycogen store is also depleted, at that time gluconeogenesis is the only pathway which can supply glucose to these essential tissues. The third important significance is that we have a protein, we have a lipid, and carbohydrate as a source of energy. But this protein, lipid and carbohydrate, all three act as a source of energy under aerobic condition. That means in the presence of O2. But when oxygen is absent, that means under anaerobic condition, protein cannot provide energy, lipids cannot provide energy, only carbohydrate, and that too only glucose can provide energy, right? So under anaerobic condition, only glucose can provide energy. And this situation occurs like whenever you are in the fasting and you do exercise. So what happens that exercising skeletal muscle, they are now under anaerobic condition and they require energy. So that energy, it can be derived only from the glucose, not from any other substance. And this glucose under fasting state, it is provided only by gluconeogenesis. The last but not the least significance is that in the gluconeogenesis, we see that these non-carbohydrate precursors, these all, they are getting converted to glucose. So if gluconeogenesis does not occur, then this precursor or metabolites will get accumulated. And some of these precursors, they are the end product of their respective metabolism. See, lactate, it is the end product of glycolysis. Propionyl coenzyme A, it is the end product of beta oxidation of odd chain fatty acid. Glycerol is the end product of lipolysis. So if gluconeogenesis does not occur, then these various metabolites will accumulate in our body and may produce harmful effect. So thank God that gluconeogenesis is occurring. That's why these metabolites are able to be cleared from our tissue. So we can consider gluconeogenesis as a clearinghouse, or it can clear the various metabolite. So it clears various metabolites. It does so by converting these metabolites to the glucose. Now let us see that how actually the steps or reactions of gluconeogenesis occur. First, I will start with the conversion of this pyruvate to the glucose. See, here pyruvate will get converted into glucose. This is one of the many possibilities of the gluconeogenesis. And we also know that from the glucose, pyruvate can be synthesized. This pathway, we call it as a glycolysis. And this pyruvate to glucose, we call it as a gluconeogenesis. So it seems that gluconeogenesis and glycolysis both are simply reversal of each other, but this is not actually true. Actually speaking, in the glycolysis, there are total 10 steps. And out of these 10 steps, 7 steps are reversible, whereas 3 steps, they are irreversible. So in the pathway of gluconeogenesis, these 7 reversible steps, they can be similar in this both pathway. But this 3 irreversible pathway, they must employ some alternate enzyme system. Alternate enzyme system must be employed to make these 3 irreversible steps in the reverse direction. Let's understand it with the diagram. This is the glycolytic pathway in which glucose is getting converted to pyruvate. Now, if we want to reverse this glycolysis and convert it into the pathway of gluconeogenesis, these 7 irreversible steps, all these black arrows, they are the reversible step. They can be simply reversed. But this irreversible step, and this irreversible step, they must employ some alternate enzyme system to make it reversible. So, by the alternate enzyme system, pyruvate can be converted to phosphoenol pyruvate. Fructose 1,6-bisphosphate can be converted back to fructose 6-phosphate, and glucose 6-phosphate can be converted back to glucose, and we can get a pathway of gluconeogenesis. So here we need to discuss only 3 reactions. First one, reversal of this, and reversal of this, and we can have a pathway of gluconeogenesis. Let's start with this pyruvate to phosphoenol pyruvate conversion, that how this reaction will get reversed. So for the reversal of this pathway, this pyruvate, it first needs to enter into the mitochondria. So suppose, let's say that this is the mitochondria. So on this side we have a mitochondria, on this side we have a cytosol. Now once pyruvate enters the mitochondria through the transporter available on the mitochondrial membrane, this pyruvate first gets converted into oxaloacetate. Now this conversion is carried out by the pyruvate carboxylase enzyme, pyruvate carboxylase. Now here there is one trick, that whenever there is a carboxylase enzyme, it requires A, B, C. A for ATP, B for biotin, and C for carbon dioxide. So first, how ATP is utilized? ATP is utilized for the energy purpose. So ATP gives an energy to this reaction. B is for the biotin. Biotin is the important cofactor for this pyruvate carboxylase enzyme, and C is for the carbon dioxide. So carbon dioxide gets added. Once oxaloacetate is synthesized inside the mitochondria, this oxaloacetate, it needs to get transported in the cytoplasm for the further reaction. So oxaloacetate is now comes out into the cytoplasm. Now actually how this oxaloacetate from the mitochondria to the cytoplasm, this transport occurs, that I will discuss in the short time. So this oxaloacetate is now in the cytoplasm. So this cytoplasmic oxaloacetate can get converted back to this phosphoenol pyruvate, and here this reaction is catalyzed by the pyruvate phosphoenol pyruvate carboxykinase, and its accepted abbreviation is PEPCK. This enzyme converts oxaloacetate to phosphoenol pyruvate. Now in this reaction, what happens? This carbon dioxide which was added to the pyruvate, it gets removed, and one high energy phosphate bond is utilized, and this high energy phosphate bond is provided by the GTP. Itself gets converted to GDP, and that inorganic phosphate that is lost from the GTP, it gets incorporated into oxaloacetate to give rise to phosphoenol pyruvate. So as you can see, there are two important things. One is that in the reaction of this glycolysis, that is phosphoenol pyruvate to pyruvate, it is a one-step reaction and one high-energy phosphate bond is generated. Whereas this reversible reaction, that is pyruvate to oxaloacetate and oxaloacetate to phosphoenol pyruvate. So it is occurring in the two-step, plus two high-energy phosphate bonds are utilized, one over here and one over here. So the reversal is a costly process. Now let us see that how oxaloacetate will be able to come out in the cytoplasm. So there are no any transporters on this mitochondrial membrane for the transport of oxaloacetate. So what happens? This oxaloacetate, it gets converted to the malate. Then this malate, it can be transported outside this mitochondria because malate has a transporters on this mitochondrial membrane. So malate comes out into the cytoplasm and it can be converted back to the oxaloacetate. Now this reaction, it is catalyzed by the malate dehydrogenase, and meanwhile NADH gets oxidized to NAD+. And here reverse reaction is occurring. So NAD+, it will be reduced to NADH plus H+. The reaction is catalyzed by the same enzyme, that is malate dehydrogenase. So remember, oxaloacetate is converted to malate just for the transport across the mitochondrial membrane, and then again it is converting back to the oxaloacetate. So once phosphoenol pyruvate is synthesized, it follows the same reaction as of glycolysis, as all are reversible reaction. Then it converted to fructose 1,6-bisphosphate. Now fructose 1,6-bisphosphate needs to get converted to fructose 6-phosphate by different enzyme system. Why? Because this is the irreversible reaction of glycolysis. So this fructose 1,6-bisphosphate can get converted to fructose 6-phosphate by the enzyme fructose 1,6-bisphosphatase. And here, as you can see, here two phosphates are there and here one phosphate is there. So that inorganic phosphate is removed with the help of addition of water molecules. Now once fructose 6-phosphate is formed, it gets converted to glucose 6-phosphate, and then this one last hurdle is that is glucose 6-phosphate to glucose. There must be some alternate enzyme system. So this glucose 6-phosphate to glucose, this conversion occurs by the glucose 6-phosphatase. And here also this phosphate molecule is removed as an inorganic phosphate, and it occurs by the addition of water molecule. So this is how from this pyruvate, glucose is synthesized. Remember that glucose 6-phosphatase, it is absent in skeletal muscle. It is not present in skeletal muscle. So skeletal muscle, it cannot carry out gluconeogenesis. Why? Because the final step of this gluconeogenesis cannot occur because of lack of enzyme. So, we have seen that how glucose can be synthesized from the pyruvate. Now, next let us see how glucose can be synthesized from the lactate. So, lactate can simply be converted into the pyruvate by the action of lactate dehydrogenase, and meanwhile NAD plus gets reduced to NADH plus H plus, and once pyruvate is formed, we know that how it will form the glucose, right. Now, see here how many molecules of pyruvate or lactate will be required to synthesize one molecule of glucose. If you examine carefully, from the pyruvate, one molecule of pyruvate, we get one molecule of phosphoryl pyruvate, then one molecule of these and these. So, we are getting only one molecule of glyceraldehyde 3-phosphate or one molecule of dihydroxyacetone phosphate. We need to have one molecule of each of this molecule. So, we need two pyruvate molecule to get converted into one molecule of fructose 1,6-bisphosphate, and then we can get one molecule of glucose. So, two molecule of pyruvate are required for synthesis of one molecule of glucose. Now, let us see that from the pyruvate to the glucose synthesis, how much energy is required. So, as you can see that one high energy phosphate is required over here, one more high energy phosphate is required over here, and one more high energy phosphate is required at this reversible step. So, we can say that total 3 high energy phosphate are required for each pyruvate molecule, and for the synthesis of glucose we need 2 pyruvate molecule. So, 3 high energy phosphate into 2, that makes total 6 high energy phosphate will be required by the pyruvate molecule to synthesize one molecule of glucose. So, indeed gluconeogenesis is the expensive, energetically expensive process. Now, we have seen that how glucose can be synthesized from the pyruvate as well as lactate. Let's see how glucogenic amino acids can give rise to synthesis of glucose. So, as you can see, this oxaloacetate and malate both are intermediates of TCA cycle. Now, so logically any intermediate of the TCA cycle can get converted into the oxaloacetate, and oxaloacetate is the boss molecule as it is a starting point for the gluconeogenesis. So, let us write down important intermediates of TCA cycle. So, after oxaloacetate, important intermediate is alpha ketoglutarate. Alpha ketoglutarate converted to succinyl coenzyme A. Succinyl coenzyme A can get converted to fumarate, and fumarate can get converted to malate. So, any of these intermediate can get converted to oxaloacetate, and then oxaloacetate can give rise to glucose. If we talk about glucogenic amino acid, most of the glucogenic amino acid, they give rise to one of the many intermediates of TCA cycle. Let's start with the alpha ketoglutarate. The carbon skeleton of various amino acid, for example histidine, glutamate, proline, glutamine and arginine. Carbon skeleton of this 5 amino acid can give rise to alpha ketoglutarate. Succinyl coenzyme A, it can be synthesized from valine, isoleucine and methionine. Fumarate, it can be synthesized from the phenylalanine and tyrosine. This oxaloacetate, it can be synthesized from the aspartate and aspargine. And one more thing, that this pyruvate, although it is not an intermediate of the TCA cycle, this carbon skeletal of certain amino acid can give rise to pyruvate also. What are these amino acid? So, pyruvate can be synthesized from the cysteine, threonine, serine, tryptophan, alanine and glycine. So, these are the various amino acids that can give rise to either intermediates of TCA cycle or pyruvate. And so, intermediates of TCA cycle can get converted to oxaloacetate, or if they generate pyruvate, pyruvate can also generate oxaloacetate, then oxaloacetate will act as a starting point for the gluconeogenesis. Now, we need to remember all these amino acids along with what type of substance they synthesize. So, for that we can prepare one trick that is based on one letter code for the amino acid. See, one letter code for the cysteine is C, for the threonine we have a T, serine S for serine. Now, tryptophan one letter code is W, alanine A and glycine G. So, you can remember CT swag based on this one letter codes for amino acid. See, pyruvate is a swag molecule, right? Because pyruvate, although it is not intermediate, it can get converted to oxaloacetate and it can synthesize. So, that way you can remember this group of amino acid. Here, in this case, H stands for histidine. Now, glutamate, its one letter code is E. Proline, its one letter code is P. Glutamine, its one letter code is Q. And arginine, its one letter code is R. Remember this one letter code, I had not made it. Actually, these are the standard one letter code. Whenever in the scientific literature, we want to refer to this amino acid, that scientific literature always refer to these alphabets for their representation. And I had already prepared one video for the one letter code for the amino acid. I had given that link in the I button as well as in the description of this video. So, if you want to become familiar with the one letter code of the amino acid, please visit that video also. Okay. So, here we have a He, PQR. This PQ are the sequential alphabets, and He that you can remember. Vim, this is already a good trick, that is successful Vim. This is valine, isoleucine and methionine. In the case of this fumarate, it can be synthesized from this phenylalanine and tyrosine. So, one letter code for the phenylalanine is F and for the tyrosine it is Y. So, first year MBBS student goes to fumarate. That way you can remember. What about aspartate and aspargine which synthesize oxaloacetate? See, oxaloacetate is the boss molecule, and boss of medical college, it is called as a dean, right? So, D stands for aspartate, that is one letter code for aspartate, and N stands for aspargine, that is one letter code for aspargine. So, you can remember dean for oxaloacetate. For fumarate, you can remember first year. For succinyl coenzyme A, you remember Vim. For alpha ketoglutarate, you can remember He, PQR. And pyruvate, as it is a swag molecule, you can remember CT swag. So, this is how various amino acid will get converted into oxaloacetate, and then oxaloacetate can carry out gluconeogenesis. So, this completes our discussion from that how glucogenic amino acid can synthesize glucose. Now, next let us see how propionyl coenzyme A, it can synthesize glucose. Propionyl coenzyme A, this is the metabolic end product of the beta oxidation of odd chain fatty acid. This propionyl coenzyme A, it first gets converted into D-methylmalonyl coenzyme A. Now, this reaction is catalyzed by the propionyl coenzyme A carboxylase. And as I told you earlier that whenever carboxylase enzyme is there, we have to remember ABC. A for the ATP, that means ATP is required over here. So, ATP gets converted to ADP and PI. B is for the biotin, that this enzyme requires biotin. And C is for the carbon dioxide, that means carbon dioxide is being added. Now, once D-methylmalonyl coenzyme A is formed, it gets converted to its isomer, that is L-methylmalonyl coenzyme A. So, this is simply isomerization reaction. So, this reaction is catalyzed by the methylmalonyl coenzyme A racemase. Once L-methylmalonyl coenzyme A is there, it can get directly converted into succinyl coenzyme A by the action of methylmalonyl coenzyme A mutase. Now, this enzyme is dependent on vitamin B12. Vitamin B12 is required as a cofactor in methylmalonyl coenzyme A mutase, and it can give rise to succinyl coenzyme A, and once succinyl coenzyme A is formed, it can get converted to oxaloacetate, and story starting from the oxaloacetate we already know. So, what happens? this propionyl coenzyme A, it can give rise to glucose synthesis. One more thing is that in case of B12 deficiency, what will happen? This reaction will be blocked. So, this methylmalonyl coenzyme A, it will start accumulating in our body. So, it gets converted to methylmalonic acid, and this methylmalonic acid, it can be excreted in urine. So, in B12 deficiency, what will happen to the urine? Urine will contain methylmalonic acid, which is called as a methylmalonyl aciduria. That is one of the important manifestations of the vitamin B12 deficiency. Many times in the university examination, this question is being asked that why methylmalonyl aciduria occurs in the B12 deficiency. So, this thing we have to keep in mind. Now, we have seen that how propionyl coenzyme A can give rise to glucose. The one last thing to discuss is that how glycerol will able to synthesize glucose. So, let us see that. So, what happens? Glycerol is first converted to the glycerol 3-phosphate by the enzyme glycerol kinase. And in this reaction, this phosphate is coming from the ATP. So, ATP gives its phosphate to glycerol and ATP itself gets converted to the ADP. Now, once glycerol 3-phosphate is formed, it can get converted to dihydroxyacetone phosphate by the enzyme glycerol 3-phosphate dehydrogenase, and meanwhile, one molecule of NAD plus is reduced to NADH plus H plus. And once dihydroxyacetone phosphate is formed, it can get converted to fructose 1,6-bisphosphate, and following this pathway, fructose 6-phosphate, then glucose 6-phosphate, and then glucose can be synthesized. So, this completes our discussion that how from the various precursors glucose can be synthesized. But one thing which is very important is that in the adipose tissue, suppose this is the adipose tissue. In the adipose tissue, there is a triacyl glycerol. This triacyl glycerol can be broken down by the various lipases enzyme. It can be broken down to the glycerol and fatty acids. This glycerol then cannot be further metabolized in the adipose tissue, so it enters into the blood and reaches to the liver, and that's how glycerol gets converted to glucose. What happens to this fatty acid? See, in our body there are two types of fatty acid. One is even chain fatty acid and another is odd chain fatty acid. Now, most of the fats in our body, they are the even chain fatty acid. This even chain fatty acid, it undergo beta oxidation, and with each cycle of beta oxidation, 2 carbons from these even chain fatty acids are removed in the form of acetyl coenzyme A. So, suppose our even chain fatty acid is a 20 carbon, then at the end of beta oxidation, we get a 10 molecule of acetyl coenzyme A. What about this odd chain fatty acid? Suppose this odd chain fatty acid has a 21 amino acid. So, this can also undergo beta oxidation, and with each cycle of beta oxidation, it gives rise to acetyl coenzyme A. Now, it can give rise to 9 molecules of acetyl coenzyme A, and by forming 9 molecules, 18 carbon will be removed, and lastly 21 minus 18, that makes total 3 carbon. Lastly, 3 carbon product remains. This 3 carbon product is nothing but this propionyl coenzyme A. Okay, and this propionyl coenzyme A, it is one of the important glucogenic substance. Now, generally in the university exam, one question is being asked that fatty acid is not glucogenic. The reason is that most of the fat in our body is even chain, so it will synthesize acetyl coenzyme A and it is not glucogenic. Second thing, even odd chain fatty acid, most of the time it will give acetyl coenzyme A, but one last molecule is the propionyl coenzyme A, which is glucogenic. So, generally speaking, fatty acids are non-glucogenic. This one is exception, that is odd chain fatty acid. So, that way we have to remember both the aspect, that fatty acids are not glucogenic, but its exception is that odd chain fatty acids, propionyl coenzyme A. So, with this I complete the discussion about the gluconeogenesis. In the next video, I will discuss about the regulation of gluconeogenesis. If you have any query or confusion, please write it down in the comment section below. Thank you.